it. So that is the thing basically. So I'll, without delaying any further, I'll just like start with the lesson base. So just please mute yourself. That's all. And any doubts, as I told you, please ask me later. Is that all right with every one of you?
Yes, ma'am.
All right. Thank you.
Yes, ma'am.
Yeah, sure.
Yes,
I'll
ma'am.
share my screen with you. So basically, human cytogenetics, when it comes to that, we are only dealing with we are going to see how the basics wherein what exactly is human and what are cytogenetics. It's genetics, hardcore genetics. And then how cell division is going to help you, what is cell cycle in relation to cytogenetics, what exactly is a chromosome, its structure, packaging, and what is the properties and function of euchromatin, heterochromatin, and so all of that comes in. So basically, human cytogenetics is the study of the structure, function, pathology, and behavior of each and every chromosome that is present in the humans, as the name implies. And again, there should be variation. If at all there is no variation, there is no evolution. We would be the same as how, hundreds of years ago, how our ancestors were, we would be the same. But that is not the case. There have been changes, there has to be changes. So is the changes taking place in our favor, or are the changes taking place, wherein, you have mutations or deletions or anything that leads to a genetic complication as such. So you've got to check that. So study of chromosomes also helps in identifying whether there is an issue or if that is going to take place. Just give me a minute, please. My only request is that you please mute yourselves as soon as you join the class. It'll be easier for you to hear whatever I'm telling. And when it comes to cytogenetics, you get to see that it involves the study of the human chromosomes in relation to disease and in health. So chromosome studies have been or are a very important part basically in laboratory diagnostic procedures right now. And then this is important during prenatal diagnosis and in certain patients with mental retardation or challenges whenever they're there. And if at all there have been birth defects earlier, and in patients with abnormal sexual development, in cases of infertility, or if there are a lot of miscarriages, all of this, that is when the genetic analysis comes into play. So when it comes to the history of it, you get to see that quite a number of people, like especially Fleming and Arnold, had actually observed the human chromosomes in the 1880s itself. From there onwards, it's like, remarkably advanced. And then you've got quite a number of achievements that have been given in your book. Basically, the year, the investigator, father of genetics, we all know it is Gregor Johann Mendel. He discovered that there is a hereditary particle which actually is being transferred from the parents. So that is how the principles of heredity came into existence. And then from then onwards, you have in the year 1867, Gustav Jamsa, very important person, you'll get to know in a little while later, invented the Jamsa stain mixture. And then you get to see that Sutton and Boveri, Walter Fleming, discovery of mitosis, meiosis, nucleus, nucleolus, chromosomes, establishment of cell lines. All of these have been there and you can get to see them in your textbook. I'm not going to go into detail because, having said that time is of essence, we were just given 10 classes for 16 topics. So you can imagine. So more or less, I will try to cover every topic, but if it is not possible, you please read. Any doubt in that matter, you please ask me. I'm always available for clarification of doubts. Is this fine?
Yes, ma'am.
All right.
Okay, madam.
Yes. And you can see in your textbook, if you can keep your textbook in front also, it will be very helpful for you only. And you can get to see that, there have been different kinds of times or periods also wherein, the discipline of human cytogenetics has evolved. You can get to see that there are 5 years: dark ages, the hypnotic period, the trisomy period, the banding era, and the molecular era. So before years 1952, it was known as the dark ages. So only it was then that the mammalian tissue culture was starting to get developed. And early studies reported that number of chromosomes in a cell were around 48. Today we know it is 46. And when it comes to the hypnotic era, you get to see that hypotonic era. I'm so sorry, it is hypotonic era. So solution of lower salt concentration was used in which cells were placed in, so that water absorption takes place, and then hypnosis, hypotonicity, that absorption of water inside. Then the swollen cells allow the chromosome to readily separate. Then it's easier to count. This was how, the correct chromosomal number as 46 was established. And we know that during the trisomy era, there are usually we know that there are a pair of chromosomes. If there is a third chromosome, that is known as trisomy of that particular chromosome number, in which again, this has led to the discovery of congenital abnormalities. For example, patients with Down syndrome. So there is, what do you call it? It is known as trisomy of chromosome number 21, which is Down syndrome. And then trisomy 18 is Edwards syndrome. So you have different kinds of trisomies, wherein there is an extra chromosome attached to that, with there's an additional copy of a chromosome, because of which there is that is, there are 3 chromosomes in that particular pair. So it's not a pair anymore. So advances led to banding of chromosomes for identification, and this brought out different horizontal bands of different staining intensity basically. Then this led to the identification and then the pattern of bands that was identified or that was made led to the identification of each particular chromosome. This in turn made made it possible for us to recognize if there was any kind of abnormality in any of the chromosomes. So again, testing for chromosome abnormality as such. So then you have right now a very high resolution banding technique that takes place, so that number of bands are visible, and this leads to a better study of the chromosomes. Now, right now we are in the molecular era, wherein DNA probes have been used in cytogenetics, so that these probes hybridized to the chromosomes and then determine a specific DNA sequence. And then if this particular DNA sequence is present on the target's chromosome or not, this is how we even again find out genetic abnormalities. So then again, in the future also, cytogenetics, more methods are being discovered and linked to molecular techniques, so that this will be of more help in the medical service as well as research purposes. Now, when it comes to chromosomes, I'll just let me share my screen with you. Just give me a moment. Yeah, somebody had asked if it is possible to send notes. I'm very sorry to say that there are no particular notes as such. I will just be able to share my PowerPoint presentations, whatever I'm showing you right now inside in the class. I will share them in your group, and ask for the important questions and the question papers, whatever. You will have to go to the university library or wherever your respective study centers are, get the questions from there, because basically we'll only be dealing with the teaching part. So the important questions, all of it, you'll have to figure it out from the previous question papers, and your project, whatever your assignments are given according to that. Done. That is what I will share the PPTs in the group. Please give me a moment. Let me share it right now. So when it comes to chromosomes, we all know that they are like the basis of DNA, and they are two in number. In higher organisms, that is, you have a set of chromosomes coming from one set is coming from the what do you call it the mother, and one set is coming from father. And okay, practical classes, I have no idea at all. You have to find out with Kishore sir. Kishore sir will be able to guide you in all of that. I'll be only teaching the theory part. You have any doubts? Again, let me tell you, please don't interrupt in the middle of the class. You can put in a message if you can, then that will be helpful. And before the class ends, I will just in the message box, put in my phone number. I think I'm already there in one of your groups, if I'm not wrong. So you can always see my name is Pranati. I think I'm there in your groups, your official WhatsApp group, basically. All right. So, right. So basic chromosomes, like I told you, they have they are the basis of DNA, basically genetic material. And you get to see that you have 23 pairs of chromosomes, that is 46 chromosomes are present, out of which 23 are you have autosomes and allosomes. We'll come to them later again. So again, you have nucleosomes, that is histones plus DNA, and then DNA is tightly wrapped around it. Each section has this DNA that's present, and enzymes can't get to the DNA unless there is an enzyme called as helicase, which helps in the unwinding of that. So a chromosome is basically a genetic material, that is DNA, histones. Chromatid is half of the replication. Basic information that you all have to know right now. These form the basis of all the human cytogenetics that we deal with. Centrioles is the centriole, which what do you call it? It helps with the organ that helps the formation of a spindle. And centromere is the center of the chromosome. So again, in prokaryotes, you get to see that you have only asexual reproduction. I request you all to please mute yourselves. You're disturbing your class itself. I think it is just one person. So anyway, so in prokaryotes you have the binary fission, and in eukaryotes you have the mitosis. So we'll come to that later. Mitosis and meiosis, that is the sexual type of division, basically. So the cell DNA or replication takes place. Division of the chromosome into parts, segregation, cytokinesis, the production of the cellular material, which you get two similar cells. Whereas in the case of the mitotic cell, you get to see that it goes through a cell cycle, wherein you have G1 phase, that is the gap of free replication phase, S phase, wherein synthesis of DNA takes place, synthesis of the genetic material, then G2 phase or the pre-mitosis phase, then you have the M phase, mitosis phase, and the gap 0 phase, GO phase, which is the non-dividing phase, which is important basically. So this is how the cell cycle is, as you can see in this picture. So again, when you want to see the chromosomes, you get to see that the structure of the chromosomes is easily visible, especially during the nuclear division when they are highly coiled. Each chromosome in the genome actually will be distinguished with a lot of several criteria that is considered. That is the length of the chromosomes, then position of the structure of centromere, then how are the two arms presence in position of enlarged areas, that is they are known as the knobs in a chromosome or the chromomeres. All these will help you in identifying a particular chromosome basically. So again, when it comes to mitosis is having 4 phases, that is prophase, metaphase, anaphase, and telophase. Again, mitosis is something wherein all the somatic cells, that is your normal cells, not your allosomes, they undergo this kind of division basically. And the function of mitosis is to see that you get an exact copy of the chromosome. There is no crossing over taking place, especially in mitosis. Interphase is also present, wherein you have it is the period between two successive mitosis that is taking place. So Again, like I told you, when it comes to mitosis, in the sense, you get to see this cell division. This is the mitosis and cytogenesis. This is the chromosome G0 or the non-dividing phase. And this is the restriction point. And then this is the cell replication in the inside S phase. And then this is where G2 phase comes in, wherein the cell division is getting to be done. So number of chromosomes, number of chromatid numbers. Chromosome numbers will be same, but chromatid numbers will double. Again. So why should cells divide? We know that there are certain cells that, it helps in, what do you call it? We have cells that are dying because of wear, tear, damage, cell death is taking place. And at the same time, many of the functions, after a certain point of time, the cell functions get redundant. That is, it has, it does not perform in the same way. So those cells definitely die. So that is when your new cells, once they start dividing, once they start growing, they come and they perform the functions again. Whenever there is division, cell division taking place, there should be accurate replication and equal division of the genetic information that's present in the DNA. So you have interphase, again, which is divided, you have G1S and G2 phases in the interphase, and mitosis, wherein, in mitosis, we have prophase, metaphase, anaphase, and mesophase. And this is the nuclear division, and this is the asexual reproduction. So you're going to get the copy of the same chromosome. So in prophase, again, you can get to see that chromatin becomes condensed. The chromosomes get condensed. They are visible. Then the nuclear membrane breaks down, and then the centrioles move to the opposite ends of the cell. So this is how it is in the case of prophase. In the case of metaphase, again, you get to see that in metaphase, they line up in the middle, and the nuclear membrane is not present anymore. And then you get to see, the beginning of prometaphase is also present. Proteins are there, they get attached to the kinetochores, and then they create these kinetochores after the attachment of the proteins. Then microtubules also help in attachment of the kinetochores.
Madam, excuse me.
Yes?
Madam. Madam, getting disturbance, madam, while taking class.
Yeah, somebody has switched on them. I can't mute them. Basically, I'm trying to see.
Host, you can mute, madam. You can mute to all.
Okay.
You can mute all the...
Mute
all option is there, ma'am.
Sure,
you can mute everyone.
All right, done. Thank you. Right. I hope this is fine. Right. So the microtubules get attached to the kinetochores. They're in the middle. Spindle fibers align in the middle, basically. So the cell's middle will have all the chromosomes lined up. This is known as the metaphase plate, middle or equator. Then this organization actually helps to ensure that in the next phase, the chromosomes are separated. These spindle fibers actually pull the chromosomes, because of which each cell, whatever is there, or each half over here will get different, the chromosomes, the halves of it. So this is what is happening over here. In anaphase, if you see, the paired chromosomes separated the kinetochores. These are the kinetochores, the spindle fibers. They pull the sister chromatids apart. And there is, because of enzymes also, this centromere breakage is there. This is how, after getting stained, this is how the picture in the cell can, you can identify it. A very good pathologist or a very good cytogeneticist can see a cell and identify if it is a dividing cell and in what phase you are able to see this particular, in what phase the cell division is going on in that particular cell. Anyway, so this helps, this anaphase helps in recombination of the kinetochore movement along the spindle microtubules. So you have, the sister chromatids getting broken apart. Now, when it comes to the telophase, you get to see that two nuclear membranes are formed again. Over here, you get to see that spindle fibers are getting, are disappearing. Chromosomes are not in the condensed form. They are getting decondensed. So uncoiling of the chromosomes is taking place and cell division is taking place. That is cytokinesis. So in cytokinesis, what is happening is the cytoplasm is getting divided. All the plasmic organelles are also getting divided. So this is what happens in the cell cycle basically. So that because of which two new cells are formed, and again interface begins. So this is how the cells look, and you can actually see this is the interface stage. And if you're able to look through a little more in detail, you can get to see condensation of the chromatids and all that. So this is one thing. See, this is the metaphase cell basically, and this is an anaphase cell. Slightly difficult, at least for me, to identify, but the picture has the slide has made our work easier. Spindle fiber formation and anaphase is occurring over here. Then this is a prophase, condensation of the chromosomes. So this is one thing. And in the case of meiosis, again, you get to see that, you have meiosis is divided into two again. Meiosis actual, wherein the sexual division or reproduction is taking place. And then again, meiosis is continued by mitosis. So in meiosis 1 and meiosis 2, meiosis 1 is wherein the actual division of the cell takes place with the formation of the daughter cells, which are different from the parent cells. And meiosis 2 is again mitosis, what you have just seen right now. So no DNA synthesis takes place between the two divisions when it comes to meiosis 1 and 2, which are consecutive in nature. But then the result is that you get 4 cells and half the number of chromosomes. So it is also known as the reduction division. I don't think I have meiosis right now. Let us see. This is all checkpoints. So you please follow your textbook and keep looking at it. It's just a matter of one page. You have so that's what. So again, in meiosis, what happens is, fusion of two cells is taking place. Production of a 2N zygote is taking place. So the parent cell, formation of chiasmata is there. Then you have prophase, and please look at the diagram in your textbook. And then prophase 1, what happens is centromeres are formed, but there is no division as such. Again, the bivalence of the sister chromatids are aligning at the equatorial or the metaphase plate. And then what happens is the sister chromatids remain together during the anaphase instead of getting separated. Now this leads to 2 daughter cells, but DNA is not getting replicated over here. And then in metaphase 2, you have centromeres division. And again, in metaphase 2 is the same as your mitotic metaphase 2. So you have centromere division and formation of 2 daughter cells. So the same thing, what happens is that it is prophase of mitosis takes a little more longer time than the prophase of mitosis in fact. So when it comes to that, the chromosomes become visible after they already are doubled. In the meiosis prophase I'm talking about, each homolog will be duplicated, and this is before the S phase. So you're going to get pairs of homologous dyads consisting of 2 sister chromatids. They are held together by proteins. What are those proteins? Cohesives. And then, they are going to align themselves lengthwise. So you're going to get a tetrad now. And this is also known as a bivalent again. And then they are held together by the chiasmata or chiasma, and they form 2 non-sister chromatids, and then crossing over takes place, which is an important step basically. So, and then you have a synaptonemal complex, which is an assembly of proteins. Just give me a minute. Let me see if at all I have that. I'm just going to have to pause the share. If at all you actually research or look into your text, not your textbook basically, but anywhere else for that matter, you're going to see a lot of the same matter that is being given in different forms. So end of the day, you get to read the same thing again and again, but what you want, difficult to get. So this is what happens is, in meiosis basically. So you get that's what they're written the same thing again and again. Chromosomes, what happens is spindle fiber formation, and then the centrioles and present over here, one at the top, one at the bottom, and then the chromosomes are over here. Spindle fibers pull them apart, especially in the metaphase, and in the anaphase, the sister chromatids separate. Spindle fibers shorten, pull the chromatids towards the pole. Free spindle fibers then lengthen and push the poles apart. And then in the telophase, disintegration of the spindle fibers occurs. So, and then this is followed by cytokinesis. Again, in meiosis, what happens is, this is the interphase. You have crossing over taking place because of homologous chromosomes over here. And then this is the formation of the daughter nuclei. But it is not anaphase, telophase is not being done over here. So what happens is, in the metaphase 1, the microtubules of the spindle fiber attach to the sister kyratochores of the homolog, pulling both the sister chromatids towards one pole of the cell. And then what happens is, this chiasmata keep the homologs attached together, cohesins keep the sister chromatids together. So when it comes to the anaphase 1 in that is meiosis, the chromosomes arms breaks down. This allows the chiasmata to slip apart. So you have dyads separating, and then again this is followed by meiosis 2, wherein again the same thing is done, but you have production or formation of 4 daughter nuclei. But again, because they're this is proper mitosis taking place, you have what do you call it, 4 daughter, but they are not the same as the parents. And crossing over is one point which is there where you have chiasmata formation in the meiosis, right? Wherein you have the non-sister chromatids have exchanged or swapped their sections. So this is known as crossing over. This helps in the fact that you do not have genetic identicality. That is, you are not a clone of your parents because of the crossing over that is taking place over here. And this is the most important step, especially in meiosis 1. Then each chromatid that has it has actually only a single molecule of DNA. So what happens is, the adjacent DNA molecules are being swapped or are being exchanged. And if this is not taking place properly, and a single nucleotide is lost or added at this crossover point, then what happens is, you're going to get a mutation occurring over here. It can be a frame shift mutation, it can be a deletion mutation, or an addition is also a problem. So then this leads to, again, a bigger problem in the future, again, once it goes on for multiple times, basically. Or else the cell division gets arrested at this particular point of time because of a checkpoint. So this is just a basic single chiasma, exchange that is taking place, but it goes on at a different level altogether all over the chromosomes. Multiple chiasmata are actually found. So in humans, the average chiasmata per tetrad is just over 2. So this is one thing. And then, assortment takes place, randomly, the assortments of the paternal and the maternal homologous genes takes place at the metaphase plate. Then, each cell produced by this meiosis has only one single homolog. And then random assortment in the humans. In your textbook, it's been mentioned that produces around 2 to the power of 23. That is around 8 crores 3,88,608 different combinations of chromosomes. That is the kind of number of what do you call it, assortment and combinations that we are just basically looking at. And further, because of this, none of the chromosomes is just only maternal or just only paternal in nature. So of all the billions of such sperms that are being produced by a man in his lifetime, what happens is no two have the same genetic content. So after fertilization, again, fertilization is basically reduction division. So the number of chromosomes are 2n, normally in any cell for that matter, but they become n during the fertilization phase, so that once this reduction takes place, there is union of the sperm and the egg, so that you again are producing the diploid. Taking these three mechanisms, that is what they have shown, the reduction division, random assortment, crossing over, what happens is the parents differ genetically, and no two human beings have ever shared. Even in identical twins also, they do not have an identical genome. Except, that is what, unless they have an identical sibling that is produced by the same fertilized egg. That is the fraternal twins. So again, behavior of the chromosomes is that 2n to n, and fertilization is like, again, n from male, n from female joins together to form the 2n. This is the basis for Mendel's rules of inheritance. Again, they have given a detailed view about leptotene, zygotene, pachytene, and diakinesis. We'll just kind of look into it. Leptotene chromosome condensation is seen, pairing of chromosomes is seen, and then separation is also seen. Then, so the non-sister chromatids recognize or shared, know that, the shared regions of DNA is not exactly certain in this particular points, and you have double-stranded breaks present. This is there in the DNA of the chromatids. Then once this is done, you have the zygotene phase, wherein, you have a synaptonemal complex that is being formed. DNA strands of non-sister chromatids begins the process of recombination. And then you have this synaptonemal complex that is being formed, which is around 100 nanometers thick. Then you have pachytene phase. Synapsis is complete over here. Recombination occurs. And then, this is going to take place so that there should be at least one pair of bivalent present, and this is where the crossover point is present. And then they have enzymes for necessary for DNA recombination and repair if necessary. Then till the end of pachytene, this is going on. Once this is done, you have diploidine, wherein DNA recombination, which started in the pachytene phase, continuing, and then you have and then the combination, DNA recombination gets completed in the diploidine phase, and then that is when the synaptonemal complex, whatever is formed, it breaks down, and then the chromatids are pulled apart. Then chiasmata takes place, wherein the recombination nodules are present. Then they migrate towards the end of the chromatids. Then diakinesis takes place, when the chromosomes decondense, and then this is followed by the metaphase 2 and the rest. In fact, now when it comes to this, I will have to tell you. Please just give me one minute. Gosh. Yeah. So when it comes to the origin of chromosomes and its abnormalities, you get to see that this process is not exactly flawless. So you get to see that almost the quite a number of chromosomes abnormalities that takes place. So when that is taking place, you get to see that like, what do you call it? It can be because of, a kind of error that is taking place. Usually like, meiosis 2 or meiosis 1 crossing over is not proper. Or, when you're doing it, like, start to share. So basic, like I told you, just a recap of whatever has been told till now. What are genes? Units of inheritance from parents to progeny. And the first two topics, at least what I will do is, it is just going to be a generalized one. You have already seen euchromatin, heterochromatin and all that, the applications part. So just like, you please go through it. I think, you will be able to understand more. So when it comes to chromosomal abnormalities, that's what extra, missing chromosomes, and structural issues such as translocation. Whatever you're reading has been not like, has not been read properly, crossing over has not taken place properly, and inversions, duplications, deletions, whatever happens. So that is when, this leads to anomalies in the chromosomes. Again, you have something called as aneuploidy, that is gain or loss of the whole chromosome, and it is one of the most common abnormality that has been seen. And then this is because of the non-disjunction. Even this chiasmata has to break. If it is not breaking, what will happen is the amount of chromosome will always be the, like, this part will just be carried over, because of which what will happen is the ploidy of the chromosome, that is, it should be 2N, is equal to 46. So that will change. So this is because of the non-disjunction of the homologs during the meiosis 1 or the sister chromatids during meiosis 2. And zygotes will miss one chromosome. This is known as monosomy. And if at all there is one extra chromosome, it is known as trisomy. And that's what, there are 3 or more X chromosomes are available, but all of them are not being, like, functional. That is also there. So to see all of this, we'll have to know the basics of chromosome structure. So let us just like have a look into the chromosome structure. So when it comes to the chromosome structure and the chemical constituents, just please give me 1 minute. So it has DNA, RNA, and I think I have to share histones, non-protein histones that are present. And if you see the structure of the chromosome, you have, it is made up of 2 identical chromatids. A P arm, which is a short arm structure. This is the centromere, wherein the 2 arms of the chromosome are actually joined together. And then this is the Q arm, which is a long arm structure. And then this is the DNA molecule, which is string-like. And that's what it's like, formed with a compact structure with histones, which are proteins. So centromere again is the localized region of the chromosome wherein the spindle fibers are attached, and chromatids are one of the 2 distinct subunits of the chromosome. So this is known as a chromatid. See, this is known as a chromatid. This is known as a chromatid. So these are the chromatids. These are the arms. P arm, Q arm. So they are sister chromatids, identical chromatids. So again, secondary constriction is wherein, what happens is some chromosomes have another one called as this, this is the primary constriction over here, as you can see, centromere. Again, another secondary constriction will present, which is called as a satellite or a trapant. So a chromosome having this satellite is called as a satellite chromosome. Telomere is ends of the chromosomes. As you can see, this end of the chromosome, it's known as a telomere, and they are highly stable and they do not fuse or unite with the telomeres of other chromosomes. Now, what is a chromomere? The chromosomes of certain species have small bead-like structures which are known as chromosomes. But the structure of the chromomeres in this chromosome is constant. As I was telling, a satellite chromosome. See, this is the primary this is the centromere. This is the secondary constriction which is present on a chromatid, and this is known as a satellite chromosome. So chromosomes number 13, 14, 15, 21, 22 have the satellite chromosomes, and they are the examples having the secondary constriction. Sorry, this is how it looks. See, this is the P arm, this is the Q arm, and this is the satellite or the secondary constriction. This is the centromere, and these are the bands, and these are the sister chromatids. This is the telomeres or the ends. So, as you can see, this is the telomere. They are highly stable. They are made up of chromatin fibers. They maintain the structural integrity of the chromosome physically, and that's what have euchromatin and heterochromatin. The chromosomes are made up of U and heterochromatin. Will come to that. So, chromosomes means thread-like structure of the nucleic acids and proteins carrying genetic information in the form of genes. What is a chromatid? Each chromosome is divided into two chromatids which are thread-like, and they contain the double helix of the DNA. Chromatin is the material with which the chromosomes are made up of basically. Other than the bacteria, they have again proteins, RNA and DNA. So this is how it is. If you see, these are the chromatids. This is like, this is a chromosome. This is the chromatin material, and this chromatin material is made up of the DNA double helix. So all you're going to do is take those chromosomes and then slowly start to open it. Just imagine you taking a wool, just like in the egg shape, start unwinding it. Finally, you'll come to DNA, just coiled. So, coiled strands of DNA bound to basic proteins, that is histones. Again, you have two types over here. One is euchromatin and heterochromatin. So, when it comes to chromatids, basic structure is basically a nucleosome. So, as you can see, the two arms are the chromatids over here, centromere over here, and then heterochromatin is densely condensed or more condensed, and then you have it is having methylation taking place and high adenine and high content, so it stains darker. Euchromatin or true chromatin. U means true, so it is less condensed. It can express the genes. It has higher GC content and it is staining lighter. This is how it is. So you can see the difference between heterochromatin and in the nuclear. This is the nuclear core basically, as you can see. So again, this nucleosome is having a solenoid wherein it has a core linker DNA. You can see the figure in your textbook as well. And you have one molecule of the histone, H1, and you have chromosomal proteins. So this is how it is. So these are the histone proteins. This is the histone core nuclear complex basically, and two copies of 4 kinds of histone are present. That is H2A, H2B, H3 and H4. And H1 is the linker DNA. So like you said, please see this particular picture. You can get to see that it's this double DNA double division, right? DNA helix. So this part is actually in the form of beads on a string of the chromatin. Chromatin material material is surrounding the DNA. That is H2A, the histone proteins, I mean. Then this is packed in the nucleosome. And this part is shown over here, this section of the chromosome. And this is again in the condensed region of the chromosome. And this is present in the entire, this small part of the mitotic chromosome. And not even near the centromere, it is just in one of the arms, that is the Q arms. So this is how it is. And again, a better picture is almost the similar picture is shown in your textbook as well. And then when you come to see, the histone also has some modifications done. So primary structure is almost similar, but when it comes to the modifications, you get to see that acetyl groups are changed to lysine groups, and phosphate groups to serines and threonines, and and methyl groups to lysines and arginines. So this modification takes place, and it says 75-80%, but you please have to put a hyphen when you're reading it. Doesn't say it is not 75-80%. There is no 75-80%. It is 75 to 80% of the histone molecule is incorporated in the pore. Please keep this in your textbook, when you're making a change. Right, like I told you, a lot of people have just kind of told that the textbooks have a lot of spelling mistakes. So in any case, you're able to configure that it is a mistake. You can do it on your own. Otherwise, you can just ask me or discuss it in your group regarding the same thing, so that everyone for that matter, it will be helpful for everyone else as well. And as I told you, whatever slide I'm showing you, I am going to share it with you in the group. So you need not worry about this. Right. So these are the kind of histone modifications that are there. And then again, you have reversible and irreversible kind of modifications taking place. So if at all the acetyl groups are, having enzymes such as histone acetyl transferases, which is known as HATs. Again, HAT medium in monoclonal antibodies is totally different. We will be coming to that hopefully later. Otherwise, don't get confused. So these histone acetyl transferases, if at all it is added to it, and then it is a reversible reaction again. Otherwise, if it is removed, then you have a deacetylase coming into action. So this is taking place in the chromatin region, and then from there onwards, gene transcription will follow. Right? So this is one thing. And then methylation helps in the neutralization of the charge on the lysines and arginines. And then methylation of lysine 4, that is the amino acid present in the DNA, is in the H3 histone. Then this results in active genes. And if there is a methylation in lysine 9 in H3, again the same histone 3, then gene inactivation takes place. And this this leads to permanently inactivated in the somatic cells. You'll read about it a little bit later. So this is the kind of changes, acetylation, deacetylation that can take place, or phosphate groups are being added or changed, and methyl groups are added to the lysine and the arginines, etc. So again, variants are there. You're going to find out this variety that is present at the linker DNA depends on the factors such as the type of cell, presence of where the chromosome is present in the cell cycle, and the stage of differentiation for that matter. And then replacement or variation takes place either by being replaced by a different protein altogether, or it can be replaced by another variant, so that especially for example, H2A is replaced by variant H2AZ between U and heterochromatid for that matter, and then so that there are changes taking place. And in some other examples include that H3 is replaced by CE, that is centromere protein A at the nucleosome area, And then if this is not taking place, then centromere structure function is not it's blocked. Then H2A is replaced by TE variant H2AZ. This is again between the euchromatin heterochromatin boundaries. Then all the standard histones are replaced by variants as the sperm gets developed. So that's how it is. Let us see if at all I have this euchromatin versus heterochromatin. Functions of chromosomes are being there. What is exactly Chromometer? Chromomere. What exactly is a chromomere? See again, they have shown chromosome, chromomeres, chromonemata, centromere. This is the satellite again. Now we'll have to come to the classification of chromosomes also. I'll just be there. So again, this is not the slide that I was looking for, the differentiation for euchromatin versus heterochromatin basically. See if it is available. Again, cell cycle, control of the cell cycle is based on the checkpoints basically. So if you see, G1 phase, G2 phase, S phase, M phase, all of them have different kinds of checkpoints. So the G1cdk Cycle. So this is again based on two kinds of enzymes, cyclins and kinases. We'll just come to it sometime. Thing is, I have around 3 or 4 PowerPoints, just open right now. And despite the fact that they are in chronological order, what has happened is, the part what I'm looking for right now, euchromatin versus heterochromatin, is missing. So right. So as you can see the screen, interphase, what happens is during the the chromatin structure gets condensed. So density of this chromatin varies at the different phases of the cell division. So the dense regions are called as heterochromatin because they are they stain darker. Light staining causes euchromatin or the euchromatin. So again, heterochromatin has two groups. Constitutive, which is always present permanently and it does not get changed to the euchromatin stage. But facultative heterochromatin, it has euchromatin so that whenever it takes staining, compactness characteristics, they change. So it can turn into euchromatin at any point of time depending upon the situation. So this is about the euchromatin and heterochromatin. And heterochromatin is found wherein there are only few genes or no genes present, such as the centromere part, the telomere part. This is made up of the heterochromatin. But euchromatin, is present wherein, it is in the parts of chromosomes that have genes in them. And that's what it is, AT rich in for that matter. And it euchromatin is the heterochromatin is GC rich. I think we have just seen that slide a little while earlier. So again, heterochromatin is densely packed with a lot of junk DNA in them, replicated only in the late phase of the late S phase, and it does not have a lot of crossing over taking place. And again, if you get to see, heterochromatin has reduced crossing over, but increased methylation of the cytosines in the CPG lands it takes place. It has high AT content that is because of which it and then methylation is increased in the lysine 9 in the histone AH3, so that heterochromatin protein 1 can bind to it at that particular place. This blocks the access for transcription factors needed for the gene transcription basically. Whereas in the case of euchromatin, again it is less condensed and it has more genes that are being expressed, and it is separated from the adjacent heterochromatin by insulators. Again, DNA which is like slightly different, and then the stain slider, and there is decreased methylations in the cytosines CPG lines of the DNA for that matter. Histones are being acetylated in this euchromatin, and there is a decrease in the methylation taking place. So this is about euchromatin and heterochromatin. When it comes to the nuclear pore complexes, see if I have that. So again, you have different kinds of ribosomal RNA that is being transcribed from and then once formed, they associate with different ribosomal proteins. All of them are synthesized in the cytosol, necessary for the mechanism of the nuclear pore complexes to function properly. So when it comes to the nuclear pore pore complexes, you have a nucleolus organizer region for that matter. So again, the nuclear envelope is, if you can see this picture, has quite a number of pores on it. Okay, and each is constructed from a number, and then you have a nuclear pores present. Got another better picture I'm trying to see. I don't think this is there. Slide. Anyway, to continue with our lesson, the nuclear pore complexes have it is an aqueous channel which connects the nucleolus and the nucleus to the outer cytosol. So the inner nucleoplasm is in continuation with the cytoplasm with the channels that are created, and these channels are almost like around 25 nanometers large, and then this helps in the transportation of whatever genetic material, that is the ribosomes that are produced, and at the same time, to keep this pore complex to be open, you need energy for that and different carrier molecules so as to transport the the RNA for that matter, and then you have docking molecules also which help in keeping the pore complex stable. So when it comes to the nucleus, there should be import and export of the substances inside and outside the nucleus. So for that matter, when it comes for us to actually import stuff into the nucleus, what happens is the histones which are necessary, they have to come to the nucleolus. All the ribosomal proteins, they have to be produced. They have to be imported into the nucleus for assembly. All the transcription factors for the genes to be turned on and turned off during the DNA replication, they have to be imported from the cytosol. And all the splicing factors, the spliceosome complex factors, whatever is necessary for the process of the pre-mRNA, so that mature mRNA molecules are produced, and to perform the splicing, that is introns and exons. Exons should be removed, introns should be kept, so that you get a readable frame as such. So all of these have to be imported into the nucleus. At the same time, for exportation, you need to see that the ribosomal subunits having the RNA proteins should be exported. mRNA molecules have to be out of the nucleus because protein synthesis takes place outside. tRNAs, that is also, and taking place outside in the cytosol basically. Transcription takes place in the DNA, that is what, in the nucleus. Translation or mRNA protein synthesis takes place in the cytosol. So this particular, for this to happen, all the tRNA, mRNA that has been used should go out into the cytosol. So export from the nucleus is taking place. So both the RNA and the protein molecules have a characteristic nuclear export sequences which are known as NES. So this is there. You can see that in your textbook, a good picture in fact for that. And then you have something called as the nucleoplasm, which is described the cytoplasm inside the nucleus is known as nucleoplasm. Again, for example, there is evidence that DNA replication transcription occurs at discrete sites within the nucleus. So one thing that they have kind of told. Now I want to go into the cyclins and the DNA checkpoints. Right. So for a cell to function properly, you get to see that there should be a kind of regulation that takes place. So it can be external regulation or internal regulation. So external regulation is because of the signals from the outside, that is the growth factors. So because of which, they include the enzymes, the hormones basically. So this increases the growth and division based on the injury, that kind of thing that takes place. And then molecules of the neighboring cell also. If at all there is a group of cells that is formed, the the neighboring cell releases signal molecules, because of which this particular cell will stop growing as such. So the growth factors, the receptor, signal transduction cascade is taking place, which in fact goes, and the core transcription factors, and then this is stopping. Whereas in the case of internal regulation, you have tumor suppressants, so that they respond to the signals from within the inside of the cell, so that there is. Oh, so sorry, you should have told yeah, right. Now you'll understand what in relation. So in internal regulation, what happens is the proteins in the cells respond to the signals, and then you have certain checkpoints, which are the stop and the go signals. So again, the cell cycle is divided into G1 phase, S phase, G2 phase, and mitosis. So what happens is in G1, you have checkpoints. So if at all there is any issue in the G1, that is basically DNA synthesis, if G1 is going on properly, then it should go into the next phase, that is G2. But there is a G1 checkpoint. So which determines if the cell should go or it shouldn't delay the division or enter the G0 phase. So G2 again, should the cell division go further? Otherwise, should it stop at this G2 phase, rectify whatever problem is going on in that particular phase? So you have a G2 checkpoint. Again, same thing with M checkpoint, end of metaphase, so that mitosis cytokinesis will go after the M checkpoint, because of which should there be a cell arrest at that particular point, so this checkpoint will tell. So this is all because of two different kinds of enzymes. Cell cycle control is because of cyclins and kinases. So cyclins are the proteins. This help regulate the movement through the cell cycle. So G1cdk cycle. So you have a CDK, that is cyclin dependent kinase. Cyclin dependent kinase complexes are there. So you have a G1cdk cyclin complex, then you have an S-CDK cyclin complex, G2-CDK cyclin complex, M-CDK cyclin complex. All of these are the CDK cyclin complexes, because of which the cell can either be arrested at that particular one, that particular phase, or the dissolution, or the CDK cyclin complex means wherein the phosphate group will come and attach itself, and the cyclin protein comes and attaches to the complex, because of which there is either breakage or attachment. This leads to the further going into the next phase as such. So as you can see, this is the cyclic dependent kinase, and this is the cyclic activated complex looks as such. Now, kinases are enzymes which activate, like, you have this or deactivates for the phosphorylation. That's what you have. You're having kinase means it phosphorylates. So a phosphate is added, ATP is broken down, energy is released, because of which what happens is, you have the complex breakage or going on ahead. But the amount of kinase is constant as such. All right. So this is all about the cell cycle with relation to the cytogenetics that take place. Again, interphase cell, what happens is you have this cell that is arrested in the G1 or the G0 or the S or the G2 phases. Now, they have given quite a bit of workup regarding the structure of the chromosomes and then, staining of them. And let us see. I will share this particular, presentation also, or at least the link to it. Some of them I have as such, as downloaded. Others I only have the links. You can just go through them. So what happens if this cell cycle does not get regulated? So that is how cancer cells are formed, proto-oncogenes, tumor suppression, and mutation. If these checkpoints are not working properly, that is when you'll have an issue with this, and then that's how cancer arises. So now again back to this PowerPoint wherein you have genes, chromosomes, telomeres. Structure of the chromosome. Chromosome is formed from a single DNA molecule having many genes, and it has 3 specific nucleotide sequences, that is a DNA replication origin, centromere attached to the DNA into the mitotic spindle, telomere located at each end, and then this is highly condensed, and it is the the human DNA helix occupies a lot of space in the cell, so that is why it is totally condensed. So chromosomes are stained basically, that is you have a certain dyes to stain the chromosomes, and then that is when you can get to see the karyotype, that is the type of the chromosomes present in a cell. It is known as the karyotype. Basic, easiest way to understand the what exactly is a karyotype. So each band has around 1 million of DNA nucleotide pairs, and this has only again corresponds to the functional structure. And when it comes to the karyotype of male, you get to see that human haploid genome, that is the N, it has around 3,000. So that is more than 3,000 million DNA nucleotide pairs divided amongst the 22 pairs of the autosomes and 1 pair of the allosomes, that is the sex chromosomes. So you have quite a bit of GMSA staining techniques basically to identify the human the chromosomes as such. So human chromosomes have 23 pairs of chromosomes, and of which 1 set come from the mother, 1 set from the father, even the autosomes. But 22 of these sets are called as autosomes or sex chromosomes. 1 set are the allosomes, which this 1 set again, female has 2 X chromosomes, male has X and Y chromosomes. So this is necessary to see or diagnose different kinds of issues such as the Down syndrome or different kinds of leukemia for the and to find the abnormalities, as you have already seen. Now let us see what a karyotype is. Like I told you, this is a karyotype. Photomicrograph of an individual somatic cell metaphase chromosomes arranged in a sequence. So over here they arrange it according to the lengthwise, from the longest to the shortest. Okay. And then once this is done, So how are you supposed to perform a karyotype? This is there in your textbook, in your second lesson basically. You can go through it later, like in what do you call it, in your second unit for that matter, which is chromosomes and karyotyping. So I'm just like kind of dealing it with it right now because like I told you, I will have to take in 2 topics at a time in a class to at least try and finish your portion, which is 16 units. So certain times clubbing of the topics will also take place. End of the day, I will be teaching you what is there in the textbook, no doubt about it, and what is there in your portion. So when it comes to karyotype performing, you can see that, the slides, you prepare slides for that matter, and then you get to see. Usually the metaphase slides are taken, the cells when they are there in metaphase. Around 10 to 30 cells are present, analyzed under the microscope. And then in that, you get to see that a photograph is taken, then analysis is done. And usually this is the male karyotype because it's XY, right? So human male sperm cells are seen for this particular. And the motile sperm cells, how many are viable, how many are not motile, that is also seen. So, and how do scientists identify the chromosomes? 3 key features, that is size, banding pattern, centromere position. Easiest way to tell the differences is the size. And then you have banding pattern. So Jamesa stain is a kind of stain that is used to band the chromosomes. That is by adding the stain to the cells, and then the different kinds of bands are shown based on the kind of absorption as such. So this technique is known as banding, and then they are spread on a slide, stained with a suitable dye. You view it under the microscope, and then a band is defined as a part of the chromosome which is clearly distinguished between 2 adjacent segments. So you have light and dark bands basically, and then you have chromosomes which have continuous series of light and dark. And then you have the centromeres also, which help in identification basically. Whereas the exact position of the centromere also makes you see or identify where that particular, what that particular chromosome is. So again, when it comes to the banding techniques, you have 2 different kinds of. So the metacentric chromosomes, you have different kinds of, based on the metacentric, chromosome centromere also, you have different kinds of, identification basically. If in metacentric chromosomes, centromere lies in the center of the chromosome. See here, like this. And in submetacentric and very submetacentric chromosomes, what happens is centromere is off center. It is not exactly in the middle, but the centromere is either on the chromosome arm that is longer than the other. And in acrocentric chromosomes, the centromere resides at the end, very end. Over here, metacentric in the middle. In the submetacentric at one of the ends, so that you have longer arms and shorter arms. But in acrocentric, the centromere is actually at the very end of the chromosomes. Now, when it comes to banding of the chromosomes, banding is because of you have bands distributed with different along the length of the chromosome, such as the G, Q, and R bands, and the stain that restrict the number of the specific bands on that particular structure. So you have, that is because of which you have 2 different groups. So stain, what happens is it restricts the number of specific band on a structure. So this shows what kind of chromosomes are present, and the C banding methods that are present, they do not help in the identification of every chromosome, but only it is useful in identification of a certain specific chromosome as such. And then you have G, A, and R bands, which are can be viewed in the light field or in the fluorescent microscopes. So that's what G banding means, Jamesa banding basically. And so that's what GMSR dye is used. So what happens is dark regions tend to be the heterochromatic regions, late replicating, and the AT-rich regions basically. And the bright regions tend to be the euchromatic, early replicating, and the GC-rich regions. So this is all about G banding. When it comes to the chromosomes, it's been mentioned over there. I will just have to come back to this again. The same slide will be shown to you in the next class also. So doesn't will be helpful for you to have this. I will share it till I get it. And then you have bright field R bands also. These bands are like, the reverse of G bands. What is happening is in the dark bands, whatever are there in the R, bright field R bands. So the dark bands have euchromatic. The light bands have the hetero. And when it comes to the fluorescent G and R bands, they are photographic negative of the bright field versions. That is reverse of the bright field G bands and R bands. So you have Q bands, which are like, fluorescent G bands basically. So there is fluorescence attached to the heterochromatic regions, which are most highly stained with the Q banding taking place. So whereas heterochromatin, that is what is mentioned about banding and staining in your textbook. And again, basic core difference between hetero and euchromatin is that heterochromatin has less number of genes and euchromatin has more number of genes. Heterochromatin has many repeats but no functional genes, and it has more of structural function. So you have basically your telomeres, and then you have the centromeres in the heterochromatic region. Euchromatic region has more amount of genes. So this is there. I'm trying to look into the fluorescent in situ hybridization part right now. So in the fluorescent in situ hybridization, right. So in the fluorescent, in situ hybridization, what takes place is this is used to identify specific parts of the chromosomes. So what happens is in this you can what you can do is you can take a DNA probe. This target sequence is also made up of DNA again. So it's a gene. So you know the exact location of the gene basically. Then whatever, if you suspect that there is an issue with that, what happens is you can label it with a hapten and you can put in a probe, label it with a fluorescent marker basically, which is a fluorophore, such as the fluorescent for that matter. And then you denature the chromosomes on the slide basically. That is, you have to do denaturation step and then denature the probe, add to the microscopic slide, and then there's probe formation of hybridization taking place, with which the complementary DNA is there. Then excess probe is washed off, and then you can get to see the fluorescently labeled fluorescent wherever it is there. So depending upon the different types of sites of hybridization. So this will help in, to see if at all there is any kind of chromosomal aberration, that is your translocation, or if at all there is any kind of deletion or addition. With this probe, it can be seen basically. So that is one thing. And then it is seen, that's what in cancer or in any for evolutionary divergence, from where it is exactly diverging from the parental genes for that matter, and in other diseases basically. And this is what is all about, the basic thing regarding the fluorescent in situ hybridization. Whereas use or purposes of the cytogenetics. So you need blood samples, so bone marrow, amniotic fluids, chorionic villi. That is, chorionic villi is present inside, once fertilization takes place. Chorion membrane, when it is formed, the chorionic villi actually helps in keeping the embryo attached to the uterus, inside the uterus of the mother. So this chorionic villi and solid tissue tumors, buccal smears are used for cytogenetics. So you take either peripheral blood smear, or then then you get to use different kinds of extracts for that matter. Mitogens are are added for that matter, and then you have initial karyotype, that is G banding taking place, and then you have cells being studied after autoradiography. Then a proper karyotype, that is a picture, a photo micro, a photograph of that particular DNA, the chromatids, the chromosomes present in the cell are taken photographed to see if at all there is any kind of discrepancy in it. So, for example, in your textbook, they have given a leukocyte culture, wherein initial is that you have taken that blood. Then in that peripheral blood smear, you have taken the leukocytes, separated them out from the other type of RBCs and WBCs present in the blood, basically. Then you add it to the fetal calf serum culture medium that is present, and then incubated for around 48 to 72 hours. Add colchicine after for 2 hours. Then you make the solution hypertonic, that is low salt concentration. Then use alcohol to fix it, and then use James's staining. The same picture is given in your textbook, figure 1.12. You can just go through it. So once you get the karyotype, you can actually see 46 chromosomes, that is 22 pairs of autosomes and 2 pairs of sex chromosomes. Then again, bone marrow is used for chromosomal abnormalities, especially in the leukemias, because that is necessary for analysis of the chromosomes. Then it has to be harvested as well. When it comes to the amniotic fluid, amniocentesis is performed. This is a slightly dangerous procedure, especially for the fetus, but the risk has come down quite a bit, provided you go to provided this procedure is very much necessary. Again, the safety factor is important, so they don't exactly recommend it unless it is very much necessary. So what happens is, in this amniocentesis, the amniotic fluid, which has a lot of squamous epithelial cells that are present, and that is taken. And optimal time for this is around 16 weeks of gestation, from the from the start of the pregnancy. And then if it is done earlier, fluid is scanty and there are only few cells, amniotic cells. Then if it is done later, that is culture takes around 4 weeks to do. So what happens is, this is done especially to find out fetal abnormalities. After 26th week, apparently, the abortion of the fetus is not done. Miscarriage is different from the abortion. So abortion is knowing that there is a fetal abnormality. That is when the fetus is aborted, removed, the pregnancy is terminated. Miscarriage can be again of different reasons altogether, wherein your this chorionic villi, or there is not proper attachment of the embryo with the mother's uterus, or there can be different reasons altogether for miscarriages, wherein the termination of pregnancy is natural, whereas abortion is something that is taken considering the the health of the fetus. Of course, there are other various social reasons as well. I'm not going into that. So anyway, so it takes 4 weeks. So intervention of may be difficult after the 16th week. So the main drawback is that it cannot be performed of amniocentesis, is that it cannot be performed until 15 to 16 weeks of gestation. That is the second trimester. And then this is again replaced by chorionic villi sampling. So that chromosome studies can take place and sex determination also takes place. Now, biopsy of solid tumors or tissue tumors. So abnormal karyotypes are seen in specific tumors. So these abnormalities, the chromosome structures, anomalies, can also be seen in the solid tissue tumors karyotyping. Buccal smear. buccal cavity is your basic mouth basically. So what happens is the buccal mucosa inside is taken and you scrape the skin or the cells from inside the buccal cavity and then you fix it on a pre-labeled slide, then add it to a fixative and then you air dry it. And then, this is used especially to see the result of bar bodies. Different stains are used to find out different kinds of presence of different either bar body or to demonstrate presence of a Y body. Smear is stained with quinacridine or examined under the fluorescence microscope. And when this is one thing, so what are the indications for human cytogenetic study? When should we perform all of these tests? So again, the common risk factor is high risk pregnancy. Whenever there is pregnancy after age 35, chromosomal abnormality becomes 1% or higher, which is quite high. So this exceeds the risk of amniocentesis also, because as it is, amniocentesis is a risky procedure. But having a kid who is genetically, having a genetic anomaly is much more, having a long-term problem as such. So this risk is it is better to perform an amniocentesis and find out if at all there are any kind of fetal anomalies. More than 90% of the amniocentesis is done in the advanced maternal age women itself, over the age 35 if there is any kind of pregnancy. And then third, so all this such kind of mothers are actually known as high risk carriers. And then the third indication is that it is seen to detect cytogenetic abnormality in the fetus if at all there have been any kind of previous trisomies or previous chromosomal abnormalities in a live child within the couple's family. So to see whether this particular child, whoever is growing right now, is that child also having any kind of anomaly as such, trisomy, monosomy, disomy. And one more thing is X-linked disorders. If at all, of course, you can always determine the gender of the child with this particular fetus whenever you're taking out, you're doing the karyotyping. And at the same time, hemophilia, Duchenne muscular dystrophy, they're all X-linked disorders. You can find out. So what happens is, it's possible, the female is a person who actually suffers the disease, but the male is only a 50% carrier. So already if there is any kind of history with this in the family, the mother is a carrier, the daughter is a carrier, but the son is the daughter will have the same problem, but the son will be only a 50% carrier. So this kind of anomaly can be indicated in the cytogenetics. So after this, basically, you have neonatal diagnostics also, wherein chromosomal aberrations can be, seen basically. And again, you have Philadelphia chromosome, and that is seen in the chronic myelogenous leukemia. You have certain other aberrations that can actually be detected only with the cytogenetics, retinoblastoma for that matter. And so if at all there is any gross abnormality in the karyotypes itself, then you can know that, okay, fine, there is some other problem. And then, you have to go for further cytogenetical studies. That is mutation analysis, chromosome, in a more in-depth manner. So this is all about your unit 1, basically, that is the history, and then what else is there? The chromosomes, the banding, staining, genetic workup, basically, what exactly the cell division is taking place. Then chromatin, euchromatin, heterochromatin. So we've just seen this. So I've just dealt with basic overview in your unit 2, that is chromosome, how it looks, what are exactly telomeres, sister chromatids, non-sister chromatids, and I've already told you the submetacentric, metacentric, submetacentric, and acrocentric telomeres basically. At the same time, there are you have see, the difference is acrocentric chromosomes. It's at the near proximal end of the chromosome. You have the centromere present. There is also one more group of classification of chromosomes based on the position of the centromere, that is the telocentric. In telocentric, completely at the proximal tip, this centromere is present, which is known as the telocentric. So you have two independent sister chromatids which are just like there. So only attached in the one end, that is the telomeric end. We have done this. Now I need to start about the classification of the chromosomes depending upon the groups, and then the karyotyping. I will do this in the next class. All right. So we are done with today basically. So today's class is over. It is 4:30. You can all leave right now. And I will be sharing these particular, the PowerPoint and the slides after some time. All right. Please don't expect it right now. It's not exactly possible. What I'll do is, by today is the first, right? Before the next class, I will be sharing. All right. Now you can unmute yourselves also. So thank you so much right now. You can all leave for the day. Sure. Thank you.